Okay, so our next speaker is Slaff. It's another guy I don't know at all, and he is going to talk to us about correcting for confounding known and unknown factors in differential in splicing analysis from RNAseq.
You have a preview of my presentation. Thank you. Hi, my name is Slaff. I'm from the University of Pennsylvania. I work with Joseph. I'm very excited to be here. Thank you very much to the session chairs and all the wonderful presentations earlier today. I'm here to talk about MOCCASIN, modeling confounding factors affecting splicing quantification. So RNA splicing refers to the processes by which a single gene can code for multiple distinct isoforms. So we can see here I have a diagram of a gene with the exons highlighted. Multiple distinct isoforms can emerge from the gene with the introns spliced out and some of the exons excluded. Of course, we've just heard a wonderful presentation about this. So RNA splicing the events can be defined and quantified in many different ways. In our view, we like to think about local splicing variations. In that sense, a splicing event is centered on a reference exon, which in this case would be the upstream exon. And it's a single source event with several downstream target exons, two in this case. And so we can define the splicing event as such and quantify the alternate events by assessing the read evidence for them. So suppose, for example, we have 30 reads for the first junction, 10 reads for the second. Naively, we could quantify psi, percent, or proportion spliced in for each of the splicing events in this local splicing variation. I'll step back and say, in actuality, the tool we developed, MOCCASIN, uses a slightly different procedure for quantifying psi. We combine a beta prior with read evidence to produce a beta posterior for a distribution of psi. But the basic idea is this. We want to quantify splicing events according to the read evidence. And so very often we'll have a study. We have samples from RNA sequencing. We want to do some analysis of these samples. And so we quantify the splicing events for them. And we'd like to see, for example, the samples cluster in interesting ways. And we'd like to learn from the way that they're clustering, identify genes which are responsible for the groupings that emerge from clustering, and proceed our exploratory analysis from there. Of course, sometimes what happens is that batch effects dominate the clustering. And so this is not great. It means that our exploratory analysis is confounded. And so what we have here are 27 RNA sequencing samples from acute myeloid leukemia patients. They were processed in 3 batches corresponding to the 3 colors in the top color bar. We quantify the splicing events using MOCCASIN and then cluster them hierarchically. And of course, what we see is, unfortunately, that this clustering is dominated by the batch effect. Here, each column corresponds to one of the 27 samples. Each row corresponds to one of the splice junctions. There are 10,000 splice junctions utilized in this clustering. And unfortunately, it seems like we're not going to learn very much unless we do something about this batch effect, except we'll learn about the technical variation, not interesting biological variation. And so, broadly speaking, there are a couple of approaches we could take to account for batch effects from RNAseq. We can adjust counts, and this is following the idea of a tool like Combat for expression analyses of gene expression, transcript expression. And this idea would be to actually adjust the counts so that we get, in a sense, batch-free or approximately batch-free splicing quantifications. And then we could use these for exploratory analyses. Another strategy would apply to differential splicing tests. statistical analyses for differential splicing between two cell types, two conditions, for example. And in that case, we could actually model covariates in the test and attribute some of the variation in the splicing data to the confounding factors. And so the approach that we've concentrated on here corresponds to the first option, because when we started looking at the landscape of possible tools, we saw that we could possibly adapt an existing tool that's used in expression studies, but there's not actually a tool that adjusts counts specifically for splicing, and splicing studies have some specific considerations that aren't present in expression studies. For example, as we saw before, splicing events are defined by multiple junctions in one event, and those junctions are, in a sense, competing for reads, and so we want to make sure that we take that into account somehow in our framework. And additionally, in an expression context, you might have tens of thousands of genes, maybe hundreds of thousands of transcripts, but we can have actually quite a bit more, order of magnitude more splice junctions than that, and so we need to make sure we develop an approach that works properly in the computational context we have for splicing. And so we developed such an approach. We call it MOCCASIN, Modeling Confounding Factors Affecting Splicing Quantification. And we can see that initially, this is the same initial data I showed before. It clusters according to the batch effect. After we apply MOCCASIN, in the adjusted data, we no longer cluster by batch. And you can see this both visually and statistically, and we can talk about those details later if you are interested. And so I want to talk about the method MOCCASIN Pipeline. It begins with a scaling step. We need to make sure that as I mentioned, we want to take into account the fact that junctions are competing for reads, and so we initially scale the read evidence for different splice events to make sure that we are working to account for different coverage in splicing events across the samples. After that, we can do sensible modeling and adjustment, and so the modeling method we use is ordinary least squares regression on log-scaled junction counts. So this ends up being, in a sense, a simple model. I mentioned before, we can talk about the read coverage at each junction, this y variable, and so we model the log of the junction spanning read counts as a linear model, including an intercept term corresponding to the known confounders, something like batch that the user would know about in input, the main variables of interest that could be like case control or cell type, and an error term. And then we can fit this model and compute corrected counts by excluding the effect of the confounding factors. We would reverse the scaling and then output the adjusted counts. In addition, MOCCASIN includes an option to account for unknown confounders. And the idea here is that you may have additional sources of unwanted variation that introduce systemic biases into the data. And to deal with these, MOCCASIN invokes an existing tool, RNASeq, that learns patterns of variation in the data that are not explained by the variables that the user input that we know about. And so we can combine all of those inputs, and for each junction, MOCCASIN learns a model and corrects it. And so the outcome is something like this, where we start with batch-affected data. We remove the batch effect and get data where we can get corrected counts, where we can hopefully learn something more interesting, more useful than the technical variation that initially dominated the data. Of course, it's not entirely sufficient to simply eliminate a batch effect. We want to know other things. For example, we want to know that we are increasing our power of detection for real biological effects, and we want to know that if we did differential splicing tests over samples that shouldn't have effects, that we don't get many false positives. We, in a sense, would like to know that if we correct for batch, that the covariance structure within the batches is not disturbed too much, so that in a sense, we're preserving all the right information that we should, and we have evaluations in progress that are showing us this, and so we're very excited to use this for our splicing studies. I'd like to thank everyone in my group, Roche Lab, BioCypher's group, for all the fantastic work they do, the great science that they do. It's really a pleasure to work with such a fantastic group and supervisor. And yeah, thank you very much for your attention.
So you're claiming that a regular batch correction effect algorithm, for RNAseq will not work if you want to study splicing. Do you have actually numbers for that? I mean, if you correct with a normal like combat thing over other methods, and compared to your methods for batch correction, will splicing be better, detected, or how do you evaluate that?
We've done some evaluations along the lines of what I mentioned toward the end. So we look at power to detect effects that we know exist in differential splicing. We look at false positive rate for differential splicing in groups that shouldn't have an effect. We look at preservation of covariant structure within batches, and in those respects, we see it at least in many cases, MOCCASIN outperforms combat, and some other methods, and others it performs comparably.
Okay.